All right, class. Today we are going to be going over Hops 1 or Hop Basics. Now, to give you guys an idea as to where we're starting and where we're ending, today we are going over the classic conceptualization, classic viewpoint of hops. Now, if we're to look at hops over the last, say, 30, 40 years, the way in which brewers have approached hops has changed drastically. Very, very drastically. And the research we're going through today and the principles and information we're going through today really is that old version of information or the old way in which people looked at hops. Now, hops historically were mainly just looked for their antimicrobial and bittering potential. That was it. So isoalpha acids and things like that were the main thing everyone was looking for, which is where we got a lot of the new varieties of hops we use for bittering. However, that all changed in the early 90s. We started seeing things. The big one that came out was Cascade, where we actually saw citrusy, piney tones actually be possible in beers. And then from that, we've seen things like Centennial, Citra, Mosaic, Simcoe, Galaxy, all these big, massive, fruity hops that we know today. And in some cases, that's all people know. And they forget about a lot of the stuff we're going to be talking about today. So today we are just focusing on the classic. We're talking about old school hop production. That's it. We're looking at alpha acids and all that kind of the bitter stuff, for lack of a better word. Well, next week, when we start progressing further into this topic, we'll talk more about essential oils and flavor compounds and terpenes and beta glycosides, all the things that are kind of modern day hop versions. But we have to understand the basics before we can get into that more advanced content. So that's today. So today we're going to look at hop biology anatomy. We're going to do a little bit of a dive into where do hops grow. We're only going to go into this a little bit because we're here to be better brewers, not hop growers. We're going to look at how hops are processed. We're going to look at alpha and beta acids and then look at isomerization. We'll finally finish off with three smaller topics: IBU and hop utilization, light struck, and the preserving power of hops. With that, we'll get started. So hop biology anatomy. Now, hops' technical Latin name is Humulus lupulus, and they are a member of the cannabis family, like marijuana. They are the closest genetic relative to cannabis, actually. However, the effects of smoking hops are not the same as the effects of smoking cannabis. Now, there are many different varietals, much like there are for almost every other plant. And they are, by volume and by mass, a relatively minor ingredient in beer, but they are one of the most impactful flavor compounds in beer. Now, hops are an annual climbing plant. They grow to about 8 meters tall, about 10 centimeters a day. To give you an idea as to how aggressively they can grow, NASA at one point was using these for CO2 scavenging studies. They found out that algae and things like that in water do a lot better job, but this is the runner-up for that, which is pretty neat. Now, the roots are perennial. They'll reach as far as 4 meters down into the ground. So these are really large plants when you actually start looking at their total mass. They are deciduous. They have male and females, and however, typically most facilities only want female plants. We'll get that in a bit. And they are mainly associated into cones. The cones may be broken down into bracteates and brachioles, and inside they do technically have seeds and they can be pollinated. Now, male plants are controversial because what would happen in a field is you'd start when you have a field that's only full of female plants, you get a very consistent flavor profile because it never changes. However, once you start fertilizing plants, you start producing new hop varietals. What will actually happen is, and for hops, it will change the DNA of the initial base plant, meaning that your cascade you once had is no longer cascade. It's cascade plus something else. This means you get large amounts of flavor variation, and more importantly, your yields decrease significantly. So you really only see male hops when it comes in facilities where hop breeding is associated. Now, you can find some male hop plants in the wild, which Southern Ontario has large amounts of wild hop plants. You can go out in the wild and find some pretty easily. Now, hops are typically used in a few different ways. We have the preservation properties of them. We have flavor, obviously, aroma, foam, and bitterness, all of which we're going to be talking about today and next week. Now, if you want to look at the basic breakdown, hops, the basic plant, has a central stig or a vine that all these different hop cones will attach to. The hop cones attach to the top, with the cone going down from there, and the cone is made up of all of these things that we call bracteoles. These are essentially the leaves of each one of the individual cones. Now, the thing that we're interested in as brewers is located right in the bottom, and that is these lupulin glands. And this is where all the alpha acids, all the beta acids, all the essential oils, all the big compounds we're typically looking for beer reside. Now, there are some compounds we're interested in this green material, but we're going to talk about that in a later course. So the lupulin glands have the resins and essential oils are the things that we're really looking for, and it looks like this. So what we see here is a magnified version of the inside of a hop cone. So what we have here is the bracteole, and then all these little lupulin glands are all located within it. This is the only part we're actually interested. This is where we see all of the flavors that we're looking for. Now, when we start looking at losses when it comes to hops, the loss that we see with absorption, because you have a dry hop material and it absorbs and expands as we add it to wort, those losses occur due to the green material. So whenever you see large-scale brewers try to make extracts and things like that, that extract effectively is just concentrated lupulin glands. So you don't have any beer losses, and you just get lots of flavor. Now, you may be thinking to yourself, why would I bother? Where that's so little volume that's lost. However, when you start looking at New England style IPAs, where 2 kilos per hectoliter is not an unheard of dry hopping rate, that can be a loss of 10% of your total volume, which is all profit. So when you start seeing a lot of NEPA, we started to see a lot of NEPA brewers start using just lupulin extracts because you're already investing so much ingredients and resource in the form of hops and malt and yeast into this beer that getting that extra little bit of profit, less loss instead of that profit, has a big impact on your margins. So this is something that you should be looking at. So we're going to now go over the basics. Now, hop growing does typically occur in between the 35th and 55 degrees latitude. Now, you can see in the image here that this is roughly southern Ontario, most large portions of the U.S., Texas and Florida are pretty much well left out, and large parts of UK and Asia. So we see most of France and England, most of Germany, Italy, they're all covered, these are all prime growing areas, especially China. Large portions of China, especially northern China, is prime growing region for hops. We then also have the tip of South America that has a large amount of growing region, as well as New Zealand, which we can see right down here. Now, this is the classic regions where hops were grown, but some of the fastest areas we see growing when it comes to hops are actually Brazil and kind of central South America. We also see larger portions of South Africa growing hops just because they can't get their hands on enough North American style hops. And we've seen mainland China, kind of more central China, right around the 25 degrees latitude, start growing large amounts of hops for their own domestic production because they can't get enough hops for their own. Now, hops cultivated in over 50 countries around the world. It's closer to about 60 right now. And there is now, and this has bounced back, about 50,000 hectares cultivated worldwide. Now, this recently was closer to 40,000 hectares. However, it's bounced back up. What had happened was that almost all hops grown around the world were historically mainly just bittering hops, the hops we're talking about today. However, once we started seeing about 10, 15 years ago, the demand for these hops just started plummeting as light lagers and things that started losing market share. You actually saw certain countries' entire hop industries collapse. And what these areas have done is they've started to plant more aroma varieties, more essential oil-rich varieties, more flavorful varieties. And we started to see that hectares start increasing all the way up. California and Oregon also has a massive hop growing population there that wasn't there previously. So we're really starting to see the amount of acreage of hops in the world increase, but in a different way than it was classically. So yeah. Now, hop cultivation requires a few different things. Fertile soil is obviously number one. Certain climate conditions are important, but we'll talk more about that, why that might be a little bit more contested next week. Sufficient amount of rain and groundwater. Hops are a very water-hungry plant. They need lots of water. And they're also subject to attack by a lot of pests and diseases. And they require a very timely harvest, which we'll talk about a lot more next week. Now, to start hops, you literally have people just going through the entire hop yard, manually making sure de-weeding, making sure everything's removed, and then getting on stilts, as you can see here on the right, to help string up the wires that go from the bottom of the plant all the way up into the trellis system you see above. This mainly occurs in the winter and the early spring. Now, once the hops stop growing, early spring or early to mid-spring, you'll have this crown of the hop plant that has about 10 to 20 or so different rootlets coming out of it. Not rootlets, but shoots coming out of it. And you typically do want to pare that back down to less than 6, maybe a bit more, usually a bit less. And you'll notice that every single one of these plants has several vines or several lines that comes out of it. Now, this is where you start seeing different hop suppliers and different hop growers kind of have certain preferences. Some facilities will say, I only want two shoots going per line coming out of the crown being the base plant. Some places will say, I only want one line coming out from the crown. Some will say, I want 4. Some will say, I want only 8 coming out of the entire thing. Some facilities will have 6 lines coming out of the same crown. Some will only have 2. There's a large amount of variety and variance we see amongst producers. And each one of these different decisions, much like how a winemaker tends to their field and tends to their plants, is going to have an impact on the plant's flavors itself and how much yield it produces and the alpha acid content and all those things. This would be the terroir, or the territory of, or the way in which the hop is produced. This has a big impact. We'll talk about this a lot more next week. And once we get kind of in late spring, early summer, this is what the hop yard's going to look like. All the hops are starting to race all the way up to the top of all the vines, and then once they get to the top, they'll start growing downward and then really flesh out in the late summer, early fall. Now, when it comes to pests, hops have large amounts of them, so we're going to go over these briefly. Now, remember, we're teaching you this to be a brewer, not a hop grower. If you do want more information on hop growing, the Ontario Hop Growers Association, the OHGA, is a wonderful organization to work with. Now, one of the most common ones and the most deadly ones for Ontario is powdery mildew. Ontario is a perfect storm for powdery mildew with this high moisture environment. We have high rain summers and things like that. So powdery mildew will take hold a little like this, and then when allowed to keep consistently go, it will end up shriveling up all the different leaves that are present on the vine, reducing the plant's ability to photosynthesize or produce energy, eventually killing the plant. This is why things like cucumbers and a lot of vine plants don't work very well in Ontario, simply because we have a really high chance of powdery mildew. Now, you can control for this with soil composition and things like that, but that's, again, not a conversation for this program. One of the other big ones is cone tip blight, and if I recall correctly, this is a bacteria or a virus. What you actually see is it starts breaking down the end of the cone. If you try to use these hops, they are not going to taste good in your beer. So if you notice this, it's best to cull the infected cones as soon as possible. Now, hop aphids are one of the biggest pests, but not a huge problem for Ontario normally. There was an event about a decade ago where we had a massive aphid storm followed by a massive ladybug storm. And that was a horrendous year for other plants that are affected by aphids. Now, if you take a closer look at aphids, they're just like little bugs like this. And what they will do is just eat all the leaf material present. Now, a lot of plants have specialized kind of mechanisms associated with aphids. Aphids have been attacking plants for millennia. So the plants actually respond to it. And the issue with this is that when when the plant responds to aphids and then ladybugs after, you end up often get what's called ladybug blight or aphid blight. And essentially, all the fruit that is produced that year ends up becoming very tannic, very bitter, very unpleasant to consume, which it really has a big impact. And when you have a big aphid problem, essentially all harvests from that year really get hindered. There's also something called the two-spotted spider mite. It's another thing like aphids. If I recall correctly, this actually eats aphid larvae, which is why it typically comes in succession to aphids. And that's all the ones we're going to cover. Now, there are lots more when it comes to things that can attack hops. However, we are here to be brewers. We're not here to be hop growers. So we're going to look into how hops are processed. Now, hops are typically harvested with what we call a wolf harvester. The wolf is just a commercial name, but it's often used kind of interchangeably, almost like Band-Aids for bandages. But the idea here is pretty consistent. You essentially take all the bines, you can see them strung up like this, and then they get brought through a large device, which then picks off all the individual cones, separates them with a bunch of rubber belts and air vents until you have just your cones isolated. So all the leaf material and branches are all removed. Now, historically, this was actually done by hand, and if you want to lose a few friends, grow some hops in your backyard and ask them to help you pick them. It's going to work. But hop picker syndrome was essentially a point where for about a week or so, the German economy would just shut down. And you'd have all these city folk go into the countryside and help everyone pick hops because it was a crucial part of society. Now, what would happen is they wouldn't cut down the vines like this. They'd often just go onto the vines and pick them off, to my understanding. But what happens is that hops actually have some properties to them that make you quite drowsy while working around them. They will knock you out to a lesser degree. So you had all these people on tall ladders trying to carry heavy containers getting sleepy, and people would die by falling off ladders. This is part of the reason why that's no longer allowed. So if you see someone doing that, that's actually a very large danger, and you should tell them to stop. That's not safe by any means. This is what hop picker syndrome causes. So we'll talk more about that next week, but harvesting time for these hops is incredibly important. It's kind of like wine, where you have a very small window of about a week or less in order to get those hops off the vine so they're in perfect health and perfect state. Too long, they start developing off flavors. Too little, and they don't have the proper flavor you're looking for. So it is very precise when it comes to harvesting. Now, once all the hops are stripped off the vines and all the other materials removed, freshly picked cones have about an 80% water content. Now, if we were to just leave these hops like this, the issue with this is that they would start to rot. They'd start producing large amounts of heat and start just rotting and breaking down. So we have to kiln them. We have to dry them out. Now, kiln drying brings in about 10% moisture content as rapidly as possible. Now, classically, you would use what's called an oast house, which you can see right here in the bottom right. So in here, hops would be layered inside of this hut, layer by layer by layer by layer, with about a small amount of distance between. A fire would be lit underneath the oast, underneath all the hops, and then, the fire would dry out all the hops on top. Now this means that the temperature inside of this oast house would be well over 100 degrees Celsius. What happens if you dry hops this way is that you start releasing a lot of the volatile compounds that you want in your beer, but if you release them before they hit your beer, they won't not possibly go in. So you see common commercial facilities use a maximum air temperature around 65 to 75 degrees Celsius. This way it locks in all the essential oils, they don't lose anything from the hops, and you maintain flavor. This is one of the biggest changes when it comes to hop producers in the last decade, and has had a massive increase in the flavor of some hops. It's why certain hops like Chinook went from being kind of boring and spicy to now having these big kind of grapefruit tones. It's because the way in which they are being processed has changed to preserve those compounds. Now, traditionally, this was done in the oast house, and it does still work. And the flavors that are preserved are more of the things that we classically associate with German-style hops. A little bit of spicy, a little bit of floral, a little bit of kind of those noble characteristics, as we'd call them. Those are the flavor compounds that are stable at those high temps. So if you are looking for more of a traditional noble variety, this is how you can potentially get that. But today, hops are dried in large kilns at lower temperatures with higher airflow to help dry them out. And that's the oast house. You can see this oast house has actually been converted into a house. That is what the oast house originally was. Now, modern oast houses also have one other element to it, is that they're actually pumped full of nitrogen gas to remove oxygen from them. If you've ever bitten into an apple and then let it sit there, you'll notice that the apple will start browning on the inside. This is essentially what will happen to hops, especially when we start adding heat. That oxidation reaction will accelerate. What this will do is it will oxidize and break down a lot of the flavor compounds found in the hops. So by removing the oxygen, we actually help maintain and preserve a lot of these very nice flavor compounds present inside the hops. So by using a lower temperature and nitrogen purging, we can get a lot more flavor compounds preserved inside these hops. We'll talk about that more next week. Now, once we have the finished hops, these will get turned into a few different products. We can use whole cone hops, which some brewers swear by. There are a large amount of operational issues with these because trying to pump liquid that contains whole hops has a lot of problems. And trying to separate them has a lot of problems. Commonly, however, they're turned into pellets, which you see right here. So these pelletized hops are essentially just the whole cone hops shredded apart and then extruded or pressed into these little pellets. And what you're seeing more and more done is you're seeing these hops turned into extracts, which you see right here on the right-hand side. And these extracts are essentially just the concentrated lupulin glands that we saw inside of the hops. And they do this with a process known as typically supercritical CO2, where you take CO2, liquefy it, wash the hop cones with it. That will dissolve the lupulin glands, remove that washed liquid CO2, flash off the CO2, and then concentrate what's left over. And that's typically a hop extract. Now, this then brings us to cryo hops, because this is something else that's coming out in the last little while. So cryo hops is something that's proprietary to YCH hop, or Yakima Chief hops, where they freeze the hops in liquid nitrogen. They then shatter them, all the hops, once they've been frozen. What they do at this point is, once they're shattered, it becomes a lot easier to separate the green material, so the portion that doesn't have the alpha acids or the flavor compounds with it, and the oil fractions, so those lupulin glands. Once they shatter, it's a lot easier to separate the two. So this then gives them two different products. The cryo hop portion is just that lupulin gland. So where normal, say, hops would be about 12 to 14 alpha if you're looking for a modern New England style PA variety, cryo hops can be up to 40, so they can be up to 4 times as strong as normal hops. This means that your losses are less because you have less green material. You're going to be able to use less of these cryo hops than you would normal. There are a lot of advantages to cryo hops. Now, American Noble is the green portion, the fraction of this process. And this has a much higher polyphenol content, usually much more delicate, much more stable essential, more classic essential oils, kind of those old school German noble characteristics, which these can be very, very nice in things like saisons or lagers or sours and things like that. So they do both have a purpose. Now that we can separate these two things out, it gives us a bit more potential, a bit more control as well. And that's what cryo hops are. Now, if you want to see some hop pickers in action, there are a few links here as to how you can actually see a harvester in action, but I'll let you guys go through those at your own accord. Which then brings us to alpha and beta acids. Now, the question here is that what we're trying to do is we need to ask ourselves what we're trying to get out of these hops. Now, classically, we're just trying to get the alpha acids out, and that's what we're going to be looking at. Now, when we look at the different breakdown products of a hop, we'll notice that alpha acids are actually only a very small portion. So cellulose and lignin, that green material, is about 40 to 50%. Protein's about 15%. There is some protein. Alpha acids are 2 to 17% based on varietal. Beta acids, we'll talk about in a bit, are 2 to 10. There's a small water content, usually around 10%. Minerals, polyphenols and tannins, lipid, fat, hop oil. These are your essential oils. Not a huge amount, but a big impact. And I want to point this out. There is some monosaccharides, some sugar. We'll talk about this next semester when we start looking at dry hop-induced hop creep and kind of increases in attenuation. But today we're just going to look at the alpha acid fraction. So we can break this down, this fraction, into a few different areas. We have total resins, essential oils, and then other hop compounds being tannin, sugars, and protein. So the hop has essential oils, resins, and then these kind of other fraction. And we'll talk about this last two next week. But for the resins, this is where the alpha acids, beta acids, and often the classic flavor compounds for hops would reside. Now we can break this down further and take a look as such. Total resins can be broken down to hard resins, which are less than 1%, not very useful for us, and from a brewing standpoint, horrifically understood. And then we have the soft resins, which is the bulk of the resin content. Now, the soft resin can be broken down into alpha acids, which we're going to talk about a lot today, beta acids, which we'll talk about a little, and then gamma acids, which we know very little about, but from all accounts appear to have almost no flavor impact. Now, alpha acids are unique, and we're going to go a lot more into this, but they're unique in the sense that we can actually, through boiling, cause these substances to go inside a solution, giving us what we call isoalpha acids, or isomerized alpha acids, a term I'm sure you've probably heard before. Now, beta acids cannot go through this isomerization process, and this isomerization process is important because these alpha acids and beta acids initially, before they've been isomerized, are not water soluble. They're oil soluble, which means that if we add these to beers, we're not going to get any extraction. We're not going to get a large amount of flavor extraction out of them. This is why when we dry hop a beer, so we add hops post-fermentation with no boiling, we don't see a noticeable increase in bitterness from alpha acids. We can see some bitterness from polyphenols and tannins, which we'll talk about next week and in subsequent lectures, but we don't see any real addition of IBUs, of bitterness from that standpoint. But we'll talk about that more in a bit. Now, alpha acids and beta acids are responsible for the most of the bittering characteristics, especially in old school styles of beer, lagers, pilsners, things like that. Now, they are a very small fraction, but they were historically the only thing we were really looking for when it came to hops. Now, there are many different varieties of hops and different kind of types of alpha and beta acids, but there are three main constitutions for both. Now, an alpha acid looks something like this. It always looks like this, not something like this. This is what it looks like. And I want to point out a few things here. The first one here is that this is what we call a hexose, or a six-membered ring. So we have 1, 2, 3, 4, 5, 6 inside of this core of the molecule. We then have this little R group here, which will change based on the subgroup we're working with. The rest of this molecule is conserved, but this part here changes. This is the alpha acid. When we look at beta acids, they have a very, very similar structure, except there's one difference. They're hexose, so we have 1, 2, 3, 4, 5, 6. They have this R group the same, and those R variants are the same as we see for alpha acids. But they have this additional terpene chain right here. And this chain right here is the reason why these molecules do not isomerize. So when we look at the differences between the two, alpha and beta acids, they are very similar. And those, I mentioned that R group, this R group does give us the three main subgroups of alpha acids. So the three main ones we see are humulone, which has the R group that looks like this. This is not important. This is just kind of for your knowledge if you choose to take it. The adhumulone, it looks like this little subgroup, and cohumulone or cohu has this side group you see on the bottom. And this is what makes the difference between humulone, adhumulone, and cohumulone for alpha acids, and lupulone, adlupulone, and colupulone for beta acids. This is the only real difference. The only core difference is this extra chain that beta acids have. Now, some hop producers have tried to modify the chemistry of hops to remove this and cleave it to turn beta acids into alpha acids, but that's something for another day. Now, alpha acid isomerization is incredibly important because this is the process that actually allows these alpha acids to become soluble. And this is the chemical reaction we see right here. So isomerization, we have this hexose ring, 1, 2, 3, 4, 5, 6. And the biggest change we see is that through boiling, through the application of heat, this hexose ring, 6 members, turns into a pentose ring, 5 members, 1, 2, 3, 4, 5. This may not seem like a big deal, but what this does is it turns this less water-soluble and not so bitter compound into a very water-soluble and very bitter compound. It's only because this reaction can occur that we actually get the bitterness associated with hops. That's the main reason. It's only just this. Now, degrees of isomerization are going to vary based on your environment and based on your pH. We see pH increase, the higher the pH is, the more we see this reaction occur to completion. On a normal brew, and there's always lots of variables associated with this, we only see this reaction occur to about a 35% completion rate. What I mean by that is, if I add 100 milligrams of alpha acid to that beer, I will only see 35 milligrams of isoalpha acid on the other side. This is just because the properties of the substance. Now, if we increase our pH, say we're brewing somewhere like in Guelph that has a much more alkaline, a much higher pH, and we don't correct for it, we will see north of say 40% isomerization or even higher. So this means one of two things: our beers are going to become a lot more bitter, or we can use less hops to attain the same level of bitterness. If I'm brewing somewhere like Victoria, BC that has incredibly soft water and often becomes quite acidic, I'm going to see instead of 35%, maybe 25 or 30%. Our beers will be a lot less bitter, or we'll have to add a lot more hops. pH is going to impact this quite significantly. So to kind of break this down a bit more, those three varieties do still hold true. So humulone is going to turn to isohumulone, cohumulone is going to turn to isocohumulone, and adhumulone is going to turn to isoadhumulone, all catalyzed by alkalinity, pH, and of course temperature via boiling. Now there are some variations on this, and I bring this up because this is when we'll talk about two terms from now. There is two different stereochemistries effectively for hops, and what this means is its structure. Essentially, if you can use myself as an example, it's the difference between the molecule looking like this versus the molecule looking like this. The molecule itself is exactly the same chemical composition. It's the same amount of carbon, same amount of oxygen, same amount of hydrogen, same amount of everything. But the structure and the way in which it's perceived is different. Or it's not perceived, the way in which it is maintained is different. This is important because these are not the trans and cis isomers of the same isoalpha acid have different shelf lives, different shelf stabilities, and different flavor attributes attributed to them. But we're going to talk about this at a later point in your course here, in your courses here at the college. Now, we mentioned this already. We mentioned pH already, but we didn't mention temperature. Now, everyone assumes that isomerization only occurs once we start boiling. However, this is very incorrect. Now, the higher the temperature, once we get to 100 degrees C, we are isomerizing. And typically water boils at 100 degrees Celsius. However, there are some breweries that are located high up in the mountains where boiling occurs at 97 degrees Celsius. They are still isomerizing then, even though boiling does not occur at 100 degrees C. Isomerization is based on temperature, not just the boiling. Boiling is a side effect of the temperature we're working with. So the higher the temperature I go for, the higher isomerization, the faster isomerization we're going to see. If I am, say, brewing at very high altitudes, those lower temperatures will actually result in a slower isomerization. Isomerization really starts around 79 degrees Celsius. And this is an important thing for brewers to be aware of. What you see with most modern day IPA production is that they're adding all of their hops right at the end of the boil, or more commonly, once they cast their wort out from the boil kettle into the whirlpool tank, where they add all their whirlpool hops. Now, the liquid inside that whirlpool tank is still near boiling, but it's not boiling. And incorrectly, a lot of brewers assume that because the vessel is no longer brewing, the isomerization is not occurring. But this is very incorrect. Isomerization occurs up to, as low as 79 degrees Celsius. So if I have my hops spinning inside that tank for, say, an hour and a half as cool out occurs, those hops are isomerizing to a lesser degree than they would be in the boil kettle for as long as they are present at that temp. So when you're using a large amount of hops in a whirlpool, that is going to contribute a large amount of isoalpha acid to your beer because the hops are above 79 degrees Celsius for a long period of time. Now, if you're a small home brewer and you have 20 liters of wort and you do this, you're probably going to see a 10 to 15 degrees Celsius dip as you're whirlpooling, which is quite significant. But if you're a commercial brewery and you have, say, 20 hex that you're casting out, that temperature is going to stay very high for the entire duration. It's going to stay near 99 degrees Celsius. It's one of these things that becomes important when you start trying to upscale recipes from a small version to a large scale version. Because as you cool out, the commercial version is probably going to taste a lot more bitter, mainly because you isomerized for a lot longer period of time because you lost less heat during your cool out. This is really important. Now, to evade this, and I recommend that you don't do this, some brewers have tried to knock down their wort temperature below 79 degrees Celsius, then whirlpool their hops, simply so that they can, produce products that are less bitter. This is commercially non-viable. It doesn't really work. There are a few brewers that have tried to do it, and as to my knowledge, every single brewer that has attempted to do this has given up on it because it was just too much. It wasn't worth it. The amount of time and effort and labor associated with doing so did not make sense. So they removed this option. Now, when we look at the three different breakdowns of hops, we typically see humulin be about 35 to 70% of the total isoalpha acids. It's going to be varietal and season and terroir dependent. Cohumulin is about 20 to 55%, and adhumulin is 10 to 15%. Now, there's one thing I want to dispel about this. This is something about cohumulin. Cohumulin for a long period of time was considered to be a harsh or more aggressive isoalpha acid. To the point where if you look at most hop readouts, they will give you the cohumulin readout to tell you how much cohumulin is there because some brewers hold this as gospel. Cohumulin is not more harsh or not more bitter. What we actually see is cohumulin isomerizes to a much higher degree. So if I have a 35% isomerization rate, I will probably see about a 50% isomerization for cohumulin, about a 20% for adhumulin, about a 20% for humulin and adhumulin associated. So this cohumulin is just isomerizes it more efficiently, which means you could just use less hops. You still see this given out on all readouts, but that's the reason why, because some brewers do avoid cohumulin like the plague. My 2 cents, I don't care what the cohumulin content is. I'm just going to adapt based on the known cohumulin content. And that's typically how you see most brewers that are, most modern brewers react. That's what it has right there. So with that, we're going to jump a bit more in depth into IBU and hop utilization. So the way in which brewers analyze IBUs is useful, but only minimally so. And we're going to explain as to why this is. So an IBU, chemically, is 1 milligram per liter of isoalpha acids. That's what an international bittering unit, or an IBU, is. Now, this has some problems. And the first, before we go into why, we have to explain where this test came from and what it was testing. IBU testing was mainly done for large-scale brewers. It wasn't done for the craft brewers in the modern age. It was done for your Molson, Labatt's, Budweiser's, Miller's, things like that. And if you look at these beers, IBUs really are the only bitter compound present. They're the only thing that really conveys bitterness. If there are other things that convey bitterness, those are usually a mistake in the product. When we look at modern day beers, or even stouts, porters, red ales, bocks, things like that, there are things in those beers that contribute bitterness that are not IBUs. Things like polyphenols and tannins that we get from dry hopping, especially for English ales or modern style beers. When it comes to some of the roasty characteristics we see in even bocks, especially in porters and stouts, those can convey bitterness, but they are not IBUs. So you start to see a lot of people start to treat IBUs differently once they realize what they actually talked about and implied. A lot of compounds that are not IBUs are bitter. Therefore, a lot of brewers have kind of stopped worrying about IBU. Now, from a brewing standpoint, they stopped. However, the consumer still uses IBU quite consistently. And it means something different to the consumer, and this is the important thing to kind of understand. An IBU, when they're looking at it, isn't necessarily how much iso-alpha acid it is from the consumer standpoint. From the brewer standpoint, it is, but from the consumer standpoint, it's not. What an IBU has kind of started to mean from the consumer is how bitter is this product? What is the net sum of all of the bitter substances that are found in this beer? And relatively, how bitter is it on a scale of IBU? That's kind of how you start to see the industry start treating IBU from a can. So if you look at a can and say it says 60 IBUs in the can, and it's a pale ale, I almost guarantee you that beer probably only has technically like 30 to 40 IBUs of iso-alpha acids in it. But it might be as bitter as a beer that contained 60 IBU straight with no other hop compounds in it. So it's 60 IBU net flavor-wise, but not analytically. And this is important because understanding the difference between those two can cause some problems. Now, when it comes to testing IBU, there are two main methods. Now, the first is a UV measurement, which we'll be doing in this program, which uses light. This test is probably the second most error-prone test I can think of in the brewing industry. To the point where if I give, say, 3 students the exact same beer with the exact same reagents and this exact same SOP, I will probably get an error rate of 200%, maybe more on it. It is ridiculously error-prone, which is why you've seen a lot of brewers stop doing it, because it wasn't actually giving them useful data. They're using their mouths to figure out IBU instead of actually the test. Now, some brewers still stick with this. They still do this test, and their IBUs are all over the place, and they don't make a lot of sense. And it's because of the error associated with this test. Now, the gold standard when it comes to this sort of testing is what we call HPLC, or high pressure or high performance liquid chromatography, which the understanding of this is beyond the scope of this course. But what you need to know is it is incredibly accurate, but also incredibly expensive. Now, if you do have the ability to get an HPLC in, this is what it will spit out. And I don't expect you guys to all understand this by any means, but what the take-home message is this. The higher the peak, the more of that substance there is. So what you can do with the program, once you've kind of isolated all the different iso-alpha acids and non-alpha isomerized acids from your beer, you can then run a program and figure out what each one of these peaks actually means, and then from that determine how much IBUs was actually, or just alpha acid content was present in the hop or the final beer. This is the gold standard way, and it's the way I'd recommend you do it if you have the option to it, and it's the way in which most hop producers are actually analyzing their base ingredients. They're not doing that previous test, they're just doing this. Yeah, and we mentioned this already, but pH has a big impact on the rate of isomerization. So if I go to pH 9, I'm going to see near 100% isomerization. pH 6 is about 60. Standard, you're looking at about 25 to 35, which Southern Ontario are usually ranking around 35%, just based on our water profile. Now, if you do want to determine isomerization rate, you can, first off, you need to have the data, usually from an HPLC. So it's not exactly typical or standard, but you can do it quite easily. All you have to figure out is the amount of iso-alpha acids in the beer divided by the amount of alpha acids present in the hops. Divide those two together, times by 100, you can figure it out. And there's a little run through of a question here you can help figure that out with, but we're not going to spend any time on it today. Now, there are also other elements that are impact utilization. Lower alpha acid hops, just due to diffusion, don't isomerize nearly as well. So if I'm using Czech Saaz, it's at 2% alpha acid, I will see a lower overall isomerization rate because there is less concentrated alpha acids in the hops. The higher the IBU desired, the lower the extraction rate. So my isomerization rate to go from, say, 0 to 35 is going to be a lot higher than it is from going from 35 to 60. I have to add a lot more hops to it. And that's mainly driven by diffusion. As well, the higher the gravity of the wort, the less utilization we're going to see. And this is something that we'll pick up and talk about next semester when we look at hydrogen bond strength and things like that. And with that, we're kind of in the last small little bits of topics. So light struck on hops is a big problem. If you've ever had Any sort of beer that comes in a clear glass bottle and kind of got a little skunky characteristic to it, or a lettuce type taste. This is light struck. We often do some experiments on campus where we'll put some campus lager in glass bottles in the window and let them sit there for about a month. And you open them up and the entire room smells like a skunk. It's lovely. But it's a really cool experiment. This can occur within minutes. And what you're detecting here is the breakdown of these isoalpha acids, and it looks like this. So what's occurring is this isoalpha acid reacts with light, and the light induces what we call a free radical, which we see right here is represented by this little dot. Now, these free radicals are also the same thing that you see some sort of people in the health food industry start stressing over, the reason why people want you to consume things that are high in polyphenol content to help scavenge free radicals. That's the same free radicals we're talking about. These free radicals will then cleave this bottom portion off the isoalpha acid. And now free radical, giving you what we see right here. So the core of the molecule plus this free radical containing small side chain. And this is where things get important, because this free radical side chain is incredibly reactive. Free radicals are incredibly reactive. It will then react and bind with a sulfur group, SH, which is very common inside of the wort. And once it does, it creates molecule 2-methyl-3-butan-1-thiol, also known as skunk. This is a similar compound to what skunks actually produce when they spray substances. And this is what's going to cause that light struck characteristic. Now, the folklore around this is such. The reason that you typically see things like Corona have a lime wedge present in them is that the portions on your palate that detect skunk are also the same portions that typically detect acidity. Detect acidity. So if we have limes present in the beer, it will help mask that skunky characteristic that can be induced. Now, other breweries get around this by using hop products, modified hop compounds where this process cannot occur. This is how Miller sells all their beer inside of clear glass bottles, but never has skunking problems. They've actually modified the chemistry of the alpha acids before they even hit the beer, so that this reaction cannot occur. Other large breweries that you see sell their beer in clear glass do exactly the same thing, and that's how they're able to do so. Yeah, it's just photooxidation. Same thing, just different ways to show it. So that's one of the biggest impacts for it. Skunking of hops is a big problem. Now, some people are very sensitive to skunking, and other people are not. I know one individual who is so sensitive that he typically wraps a napkin around his pints when he's drinking them on a patio, simply because he doesn't want the skunking reaction to occur, because he will taste it quite rapidly. He works in the beer industry. He used to work for a large brewery. He's very familiar with the compound. He's been tested for it. He knows this isn't just in his head. Now, we'll also look at the preserving power of hops, just very, very quickly. We're going to talk a lot more about this next semester. We'll look at different contamination organisms and things like that. But the way in which hops originally made their way into beer was actually what we call a gruit, which was the main spice mix that was originally controlled by churches and then produced by people, and then eventually went from being partially composed of hops to being effectively just hops to now being hops. Hops have an amazing preserving power to them. However, the preserving power is that isoalpha acid fraction. If we don't have isomerized alpha acids, we're not going to get preserving quality of the hops. Now, most organisms, we're talking things like E. coli, Staphylococcus, Enterobacter, things like that, things that are damaging to you, will die around 10 IBUs. But so will things like Lactobacillus, Pediococcus, all of these organisms that will, we don't want in beer, aren't harmful, but are annoyances. They will also be inhibited around 10 IBU. So if you are, say, finding yourself brewing where resources are scarce and you're just trying to preserve your beer, you're typically shooting for about 10 to 14 IBUs to make sure that beer is shelf stable. That's a really nice layer. You don't have to make IPAs that are 60 plus IBU. Shoot for about 14. You'll probably get about 12, and by the time some of those IBUs drop out, you'll probably be around 10. 10 IBUs is that golden point, but we'll talk more about this next term and in third semester. And with that concludes today's lectures on hops. Again, if you have any questions about this, please send me an email, either to the college or personal, and we'll be setting up the crowdcast for you guys to ask questions live in a few days. Hope everyone's staying well, and take care. Cheers.